perhaps in questions we can come back to specific things. So as Dominic mentioned, at the moment treatment is basically aimed at treating the symptoms. There aren't curative treatments, and so the treatments are aimed at treating symptoms like epilepsy or heart disease with kind of conventional medications, potentially modifying diet and using sort of cocktails and so on. In terms of experimental treatments, if any of you are members of the US version of Amdf, the United Mitochondrial Disease Foundation, in their newsletter after the last conference, I think they said that in the last 10 years, or 10 years ago, when they had a conference, I think there was maybe 1 or 2 drugs that were being looked at for treating mitochondrial disease, and now there are about 30 drugs that are being looked at in preclinical trials, so in cultured cells or in mouse models and so on, or some of them in clinical trials now. So again, the area of trying different drug approaches to treatment is changing pretty quickly. And some of the approaches are things, as Dominic was saying, that mitochondria are generating energy. A bit of a side effect of that is that we, as we use oxygen, we constantly spew out, if you like, a little bit of toxic oxygen species, free radicals or reactive oxygen species, and in some patients with mitochondrial disease that seems to be increased in amount, and so one approach has always been to try to use antioxidants to correct that. The problem is that conventional antioxidants don't necessarily get into the mitochondria very well, so there's been a number of approaches of trying to use antioxidants that are targeted to the mitochondria, things like Mito and I guess the EP743 or whatever is perhaps acting in that way as well. Another approach is if your mitochondria aren't working well enough, maybe if we could increase the number of mitochondria per cell by 20% or 50%, then even though they're not working so well, that may lift it up so that you've got that energy reserve, if you like, to keep things working. And so there's a number of drugs that try to target our basic understanding of what controls mitochondrial numbers, drugs that are targeting those pathways controlling mitochondrial numbers, like resveratrol and bezafibrate, to try to push up the numbers of mitochondria and see if that boosts performance. Dietary bypass therapy, again, I think for the sake of time I might stop just after this next slide or so, and then we can talk about any of these things in questions, so if there's time for some questions. But basically, fat and sugar are metabolised differently, and for some problems in the respiratory chain there's a logical basis to increase the amount of calories you get from fat so that you can bypass where the specific problem is in the energy generating pathway. That's not a general thing, but in some of the disorders, changing the calorie balance around can be logical. And recently there's also been some approaches to sort of genetic bypasses that, for example, the first part of the respiratory chain, complex 1, has 44 subunits. Used to have 45, but one of them we got wrong. So now there's 44, and there's a whole bunch of other things required to put it together. So if you were thinking of a gene therapy strategy, for example, to try to fix complex 1, then it's 50 or more different strategies. It turns out that in yeast and some other species have something that does most of what complex 1 does, but with only one protein. And so there's a whole lot of strategies trying to use that yeast protein to bypass complex 1. And similarly, there's something that does the same thing for complex 3 and 4. So these are genetic bypass strategies where we think that using these one proteins, we might be able to correct some of the problems. Mitochondrial gene therapy is the idea of, if you like, many patients have some healthy mitochondrial DNA and some mitochondrial DNA with a change, a mutation that's causing disease. And so the idea of mitochondrial gene therapy is, wouldn't it be good if we can target the disease, the mutant mitochondrial DNA, and chop it up and let the healthy mitochondrial DNA replace it. And one of the most exciting papers in the last year or two has just come out in Nature Medicine with an approach doing just that. At the moment it's sort of proof of principle stage. So it's an approach using a new technology called TALENs, where you can target any piece of DNA and basically try to chop it up. And so they showed that they can target the common mitochondrial DNA deletion or some of the point mutations and chop up the mitochondrial DNA. So that's very promising, but again, to get it into clinical use, there's a way to go. It has to, the efficiency has to be improved. We have to see that there's no safety issues around it, I guess it has to be demonstrated that it really works in a larger scale, and it's only going to work for people, I guess, where there's a mixture of healthy mitochondrial DNA. There's a bit of a concern that if it's all mutant mitochondrial DNA, then this approach would chop it all up and there'd be no mitochondrial DNA left. So clearly it's one of these things that we need to look at in terms of the safety concerns. So the other area is improving mitochondrial quality control. So in much of the time, changes in the mitochondrial function lead to mitochondria that don't work as well, and sometimes in our body we're constantly turning over mitochondria, and sometimes that's turning over one or two proteins, and sometimes that's turning over whole mitochondria at once. And there's some promising data on trying to up those pathways that recognise the poor quality mitochondria, so that we're enhancing that turnover and, if you like, boosting the average quality. So those are probably the areas on treatment. I think just because of time, I might stop there and we can take some questions for Dominic and myself, because I think we were supposed to be breaking at 1:45 or so. Yeah, 22. So I'll stop there, and I think if you have questions for Dominic or me, we can do those now, and obviously we can talk individually during the rest of the day.